Okay, this is just going to add some more context into what we were talking about last time about tandemly repeated DNA and satellite DNA. We talked a lot about it happening in the extragenic regions of the genome last time, except for when we were talking about Huntington's, which was similar to this within coding regions. This is just going to add some more context to what can happen if either entire genes are repeated, like we mentioned before with the interspersed genome-wide repeats and the implications that that can have on organisms. So last time when we talked about Huntington's, that's an example of tandemly repeated DNA within coding regions affecting the phenotype of an organism. So if you have the tandemly repeated CAG mutation, either repeated 10 to 35 times within the Huntington's gene, you're going to have the phenotype of producing the dysfunctional Huntington's protein, and you're sadly going to be a sufferer of the disease. If you have the interspersed genome-wide repeats, you're going to have one of these things that we're going to talk about now. So coding DNA can also be repetitive. For example, histone genes, which lack introns and their mRNA lacks a poly A tail, they can be repetitive, so we can have multiple histone genes interspersed throughout the genome. And the more of them we have, the more histones are going to be produced, because more of the histone genes are going to be transcribed, more of them are going to be spliced, more of them are going to be translated, and more of them are going to be folded into our histone proteins for DNA packaging, like we talked about in the first one on this playlist. Replicated coding genes can confer a survival advantage to organisms, and so it can be naturally selected. One of these examples, so cancer patients, I believe this is mainly used in leukemia, can take a medicine called methotrexate, which has the function of inhibiting a gene called DHFR, and the DHFR gene codes for an enzyme involved in nucleic acid synthesis, which is obviously important for cancer cells because they need to be able to synthesize nucleic acids to repeat. So the methotrexate inhibits that, prevents the cancer cells from replicating, and basically subsides the effects of cancer, because cancer is just unregulated cell division. The cancer cells can't divide, so you're not getting the problems of cancer. But the problem is that cancer cells are able to do a lot of stuff with their genome, and they're able to replicate like large portions of it. And some of these cancer cells are going to have replicated DHFR genes, so they're going to have this interspersed genome-wide repeats. So the DHFR gene is going to be transcribed and then reverse transcribed and reintegrated somewhere else in the cancer cell's genome. So it's going to have multiple versions of this DHFR gene. The more versions it has, the more of the enzyme involved in nucleic acid synthesis the cancer cells are able to produce. If they have enough of the DHFR genes to counteract the effects of the methotrexate, they're going to have enough of the enzyme to actually synthesize nucleic acids and replicate. And initially, the methotrexate is going to kill most of the, it's going to result in most of the cancer cells just not being able to do anything because they can't synthesize it. But the very few cancer cells that have these replicated DHFR genes are going to be essentially naturally selected because they're able to replicate and once they replicate, you have more cancer cells that unfortunately have these multiple DHFR genes to produce enough of the enzyme to counteract the methotrexate treatment, and eventually the methotrexate treatment isn't going to do anything because your cancer cells are essentially resistant to it because they were able to replicate the gene to produce enough of the nucleic acids to essentially ignore the amount of that the methotrexate is inhibiting. I hope that made sense. So that's why the treatment will subdue the spread of cancer for a time, but it won't cure it unless the residual cells with the multiple DHFR genes are killed through some other means, like say chemotherapy. But I don't think a lot of patients get them both at the same time, so I'm pretty sure the methotrexate causes some other problems, but you'll have to look into that yourself. Then some frogs can also have interspersed genome-wide repeats affecting their phenotype. They can replicate ribosomal genes and then have them tandemly repeated next to one another. So the entire ribosomal gene, it gets transcribed, it gets reverse transcribed, then it gets reinserted into the genome next to it. Actually, it may be the result of a replication slippage. Maybe. Well, the same thing would have to happen for it to be reintegrated. It doesn't particularly matter. The main takeaway is that some frogs can amplify their ribosomal genes, have them arranged next to each other, and because they have a lot of these ribosomal genes, because the genes have been amplified and replicated, the frogs are able to meet high ribosomal demands in early life stages because they're going through metamorphosis. They need a lot of ribosomes to synthesize a lot of proteins to be able to go from a tadpole to a frog. I thought that was going to be a 2-3 minute video. It's ended up being 8 minutes. I hope that kind of clarifies stuff. So the last one, we were talking about these little kind of satellite tandem repeats in coding regions and non-coding regions. If tandem repeat in a coding region, something like Huntington's, interspersed genome-wide repeats is more like the methotrexate for the DHFR for the cancer cells, or ribosomal genes in the frogs, or the histone genes in whatever you're talking about. Okay, that's enough for that. Thank you.